Good day, esteemed colleagues. A warm welcome to my talk, and thank you so much for the invitation to speak here today. It is an honor. I'm joining you virtually today from South Africa, and my name is Cloete. I'm based at the UNESCO UNISA Africa Chair in Nanosciences and Nanotechnology at the University of South Africa and the ETMA Laboratory for Accelerator-Based Sciences at the National Research Foundation in South Africa. Today, I will be discussing with you the following topic: nanofertilizers for sustainable agriculture. But first, an overview of my talk. I will be discussing with you the wicked problems the agricultural industry is currently facing, linked with how research and innovation can solve the problem in the form of novel fertilizers under the umbrella term agro-nanotechnology, its pros and cons, and what the data is telling us for future implementation. But first, a bit of background. When tackling wicked problems, a multidisciplinary approach involving agronomists, plant physiologists, chemists, biologists, and nanoscience experts are important when handling basic and applied research, innovation and scale-ups, IP and publication management, and interaction with various stakeholders such as fertilizer companies, farmers, and policy holders. Now focusing on the problem. The agriculture and agro-production industry are facing numerous and unprecedented challenges that present a threat to sustainable agriculture and food security. Abiotic and biotic stresses, including climate change, environmental pollution, urbanization, pests and diseases, the unsustainable use of pesticides and fertilizers, as well as various other factors, including multi-nutrient deficiency and low nutrient use efficiently, are significantly affecting crop yield via multidimensional avenues. For example, climate change is triggering drastic fluctuations in precipitation and temperature that are accelerating the melting of permafrost that significantly affect agricultural production parameters such as water quality, soil moisture, salinity, and fertility levels, as well as nutrient homeostasis and microbial biodiversity. In a broader context, the aforementioned parameters are also critically important for ecosystem functioning in terms of nutrient cycling and plant immunity against pests and diseases. Another example includes anthropogenic and geogenic pollution caused by the augmented use of chemical and organic fertilizers, as well as the unsustainable processing of agro-waste, as well as the ongoing Ukraine war triggering shortages in fertilizers. Such factors are systematically degrading natural resources such as soil and water, and also causing eutrophication of soil and water bodies. Essentially, by the year 2030, the demand for agricultural products will have to increase by 60% to sustain the wealth and well-being of a rapidly expanding population estimated to reach 8.6 billion by 2030. Alternative approaches to boost agricultural crop performance are hence urgently warranted. Such a solution should also deviate from the unsustainable model of increasing agricultural land input doses and, for example, energy, water, pesticides, and fertilizers, or the increased exploitation of natural resources to promote robust crop performance and production. To find solutions to the current crisis within the agro-production sector, nanotechnology have been suggested and promoted as a novel and innovative agricultural technology. More specifically, nanoparticles in the form of nanofertilizers have started to show great promise to sustainably increase crop performance by increasing nutrient use efficiency and decreasing nutrient losses. Nanofertilizers can also be designed for smart nutrient release, whilst their unique physicochemical parameters present a more efficient approach to optimizing plant nutrient uptake. Yet few studies have focused on studying the effect of nanofertilizers on seed development and nutrition, one of the most important stages of plant development. But first, let us compare the differences between traditional and nano-enabled or nanofertilizers. A substantive proportion of up to 50% of crop production depends on natural or synthetic fertilizers, of which the demand will have reached 202 million tons by 2020, further supported by a significant energy, economic, and environmental cost implication. An unbalanced concentration of nutrients within an environment may lead to soil chemical changes that directly affect soil ecology and specifically the profile of soil microbes important for the sustenance of plant health and vitality, as well as the spread of weeds and pests. Alternatively, edaphic, including phytolytic, hydrolytic, and microbial processes may volatilize or chelate and concentrate nutrients within the ecosystem, compromising its availability to plants. In effect, ultimately, up to 40 to 70% of nitrogen, 80 to 90% of phosphorus, and 50 to 90% of potassium may be lost in the environment. Benefits of nanotype fertilizers, on the other hand, include the use of smaller quantities, hence reducing transport and application costs, the provision of smart nutrition due to its targeted and slow release kinetics that could be synchronized with crop nutritional needs, its higher nutrient bioavailability, sorption, and mass transfer capacity due to its unique and versatile physicochemical properties, including small sizes, high surface to volume ratios, and increased ionizing power that ultimately increase crop productivity with limited product loss and leaching into the environment. Due to these properties, the nutrient use efficiency of the crop, defined as the maximum dry matter produced per unit of nutrient acquired and translocated into shoots and leaves, can be successfully increased to up to 20 to 30% to promote environmentally sustainable crop production. So let's look at the study questions. Before we delve into applying nanofertilizers for sustainable agriculture during a time of climate change, we need to ponder about a few questions that have not received a lot of attention in the nanofertilizer literature yet. First, how can nanofertilizers be synthesized using safe and green methods using waste products? What are their physicochemical characteristics? And lastly, what effect do nanofertilizers have on seeds towards developing sustainable agricultural practices? This brings us to the aim of the current talk. First, to phytoengineer nanofertilizers and specifically macronutrient nanofertilizers with green nanochemistry and test their effect on seed physiological parameters, with the specific objective of describing the synthesis of the nanoparticle nanofertilizing using agro-waste, to characterize their physicochemical properties and test their multidimensional effect of seed germination of the common bean or the pinto bean. So let us now focus on the methods. First of all, let's focus on agro-waste as a starting material for nanomaterial synthesis for its application in nanofertilizers. To fulfill this increasing nutritional demands of a growing world population, significant amounts of agricultural waste are being generated. Agricultural waste encompass residues from diverse agricultural sectors, such as crop residues that may include leaf litter, seed pods, stalks, stems, and weeds, and also agro-industrial waste such as fruit peels and pulp. So waste from this largest industry in the world is presenting as an environmental threat, as its unsustainable processing is polluting air as well as aquatic and terrestrial ecosystems. Ultimately, annually billions of tons of agro-waste are produced that are unsustainably processed without recognizing the easily accessible, environmentally friendly, and also cost-effective resource for high-yield new nanofertilizer development and production to promote sustainable agriculture. Nutrients that may be lost within the biosystem are also replenished, and since nanomaterials are synthesized from biogenic sources containing bioactive compounds, the potentially hazardous effect of nanomaterials inside living systems may be modulated. So in sum, synthesizing nanofertilizers from agro-waste defocuses the traditional volume principle within the fertilizer industry to a value principle in delivering a nano-sized product that can be bioengineered to exhibit a unique physico-chemical profile to fit a particular purpose. Now, Vitis vinifera is one of the largest crops in the world. A lot of organic byproducts are produced, however, with limited applications in nanosciences. To limit the effect of agricultural waste on the surrounding environment, innovative approaches to handling waste products for sustainable agriculture is warranted, and Vitis vinifera waste products have, however, had limited application and has hence huge potential for application to be processed as a nanofertilizer. So if we look at the synthesis process of the nanofertilizer, a phytosynthesis approach can be followed, and this entails synthesizing safe and biocompatible nanomaterials. This has also received far less interest in recent years, but is now growing in popularity. And only a few papers have described the effect of phytosynthesized nanofertilizers on plant growth. So during phytosynthesis, plant phytochemical compounds also have a value-added benefit in that they have diverse nutritional and bioactivity potential that contributes to the bioengineering of smart nanomaterials with unique physico-chemical properties. Furthermore, various studies have shown the potential of plant phenolic compounds that are also popular for their antioxidant activity to serve as potent stabilizing agents during nanomaterial synthesis. So if we look at green nanochemistry, the green nanochemistry or phytosynthesis approach basically involves mixing an aqueous solution of plant extract with the precursor, which is normally a chemical precursor, to synthesize nanoparticles that are collected via centrifugation and characterized then using various techniques such as transmission and scanning, electron microscopy, X-ray diffraction, and also various other techniques. So if we look at the seed treatment in more detail, specific concentrations of the nanofertilizer were selected based on analysis of the selected literature. The seeds can be imbibed for an optimal period, including 8 hours or sometimes up to 24 hours, and then various seed physiological parameters such as water uptake and growth rate are studied. So let's look at potential results and discussion of studies that focus on phytosynthesis of nanomaterials and then its application as a nanofertilizer. So basically, to visually assess particle macro and micromorphology, light micrographs, as well as SEM and HR-TEM electron analysis were completed with the associated micrographs depicting these images. The light micrograph of these types of nanoparticles shows that they are yellow clumped powder, and this specific nanopowder presents a calcium-based nanofertilizer. The scanning electron micrographs show well-formed and dense nanograins with a heterogeneous size distribution of spherical shapes in combination with nanoparticle clusters. Some nanograins are shown to agglomerate as nanoclusters due to their surface energy. EDX analysis was also performed to assess the elemental content and also purity of the nanofertilizer, and this revealed high purity of the nanoparticles that form part of the nanofertilizer. The HRTM images of the nanograins and singular nanograins are also depicted, and the size distribution of the particles is also provided in the histogram in that inset figure there, showing that the particles had an estimated average size of 25 nanometers. Particle size distribution was determined with the analyzing image software ImageJ. Then XRD patterns are also shown here, and the XRD patterns of the synthesized nanocomposites are also presented, with each following the diffraction peaks of calcium hydroxide, consensus to the JCPDS database, and corresponding to Portlandite with its crystal lattice. So if we look at the plant physiological and more specifically seed physiological parameters, in this specific study, we could see that seeds imbibed with a certain concentration of nanofertilizer showed a consistently higher water uptake compared to a higher concentration of nanofertilizer and seeds imbibed with only water as the negative control. The nanofertilizer also had an effect on the stem, root, and leaf length as well as leaf number, were higher in the treated plants than in the control treatment. Then we also saw that the nanofertilizer had a significant effect on seed germination, and this was also shown at specific or seen at specific concentration of the nanofertilizer. And we could also notice that the nanofertilizer treated plants had a significantly higher leaf area compared to plants that was treated with only water as a control. Then also further statistical analysis with hierarchical clustering showed some similarities and differences in the amino acid content between treated and control seeds, showing us that the nanofertilizer also had an effect on the seed amino acid metabolism. The color legend here of the heat map indicated a lower average amino acid concentration in red to the higher average amino acid concentration in green in a particular sample. The highest dissimilarity to average values are here shown as extreme XZ values, as shown by the brightest colors of the color map. And this was observed for the Z scores of both treated and control samples, but only for specific amino acids. Then we also determined the uptake of the nanofertilizer. This is particularly important to see the location of where nanoparticles may accumulate and cause toxicity in plants. These elemental maps determined with the ion beam technique showed that the nanoparticles representing the nanofertilizer seem to concentrate within the seed capsule. Nanoparticles with a mean diameter between 20 and 50 nanometer has been shown in the literature, and also carbon nanotubes that they can easily pass the cell wall via either active or passive diffusion or other different plant uptake pathways. Alternatively, functionalized nanoparticles may enable pore size enlargement or the induction of new cell pores, whilst nanoparticles may also penetrate and be absorbed via binding to carrier proteins, aquaporins, ion channels, endocytosis, and also membrane transporter cells. So as can be seen here, seed uptake seems to be mediated by the seed hilum, from where the nanoparticles can affect various plant physiological processes, including the plant ionome, its elemental content, its amino acid metabolism, water uptake, starch breakdown, and also hence overall germination rate. According to the literature, the mechanisms behind growth promotion have been studied at the physiological, biochemical, and genetic level and are quite diverse, ranging from directly enhancing water uptake, physiological, and also growth parameters. At the very initial stages of plant development, nanofertilizers have also shown tremendous potential. For example, seed germination is a very important and sensitive phase of a plant's life cycle, which facilitates seedling development, survival, and population dynamics. However, seed germination is affected by a range of environmental, genetic, and soil, water, and chemical factors, with seed dormancy being a significant problem leading to crop yield reduction. Since nanofertilizers catalyzes reactions at the molecular and metabolic level, triggering enhanced water uptake that ultimately leads to enhanced growth parameters, they may find significant applications as a seed germination agent. So, in conclusion, this work describes the first report on the use of grapevine leaf aqueous extract as a reductant, a chelating agent to green synthesized calcium hydroxide nanoparticles. The calcium hydroxide nanoparticles were characterized using various techniques. The data showed well-formed, mostly spherical nanoparticles. Molecular modeling showed that plant phytoactive compounds interact with the nanoparticle surface, stabilizing it, and the nanoparticles significantly affected seed germination by increasing water uptake, starch degradation, amino acid content, and ultimately plant growth. In effect, the obtained nanoparticles may serve as a nanofertilizer to improve seedling development. In sum, since nanofertilizers can be synthesized from agro-waste, presenting a circular economy using facile and environmentally friendly methods, enhance water uptake and plant growth parameters, the potential of nanofertilizers to promote seed germination and ultimately crop performance during a time of climate change that are significantly impacting soil nutritional quality hold great potential. Still, many questions remain, with specific emphasis on nanotoxicology. More interdisciplinary research involving stakeholders from the different agricultural industries are warranted to answer these complex questions in finding a viable solution to the challenges in terms of fertilization the agricultural industry is experiencing at present, and nanofertilizers can really revolutionize agriculture. Thank you so much for your attention.